My name is Tamara Mammon, and I am a junior in agricultural and biological engineering. This summer I was doing the Dury internship where I was studying the influences of overwinter temperature on fish reproduction. And what we're running today is a fatty acid analysis. And what we're going to do is grind the samples, and then after we grind them, we put them in these tubes over here and add the needed chemicals, and then they'll sit overnight so we can process them further the next day. And the reason fatty acids are important in fish samples is because once you have the different levels of each fatty acid within the fish sample, you're able to do other calculations to get, say, their protein content or carbs, other body compositions. And once you have those, you can then determine the total energy content within the fish and where they're allocating all their energy. This vial right here is a muscle sample of a yellow perch, and what we're going to do is kind of cut out half the muscle sample into a clean container. Okay, so that's a good amount. We always want to leave some in case our samples don't go the way we want them to, so then we can rerun them. And then I'm going to grind it up. So once I'm done grinding, I'm going to take a pre-cleaned weigh tin and go over to the scale to weigh it. So initially I'm going to put the pan on the scale and zero it. Then I'm going to go ahead and add about 0.5 grams of sample into the tin. So now that we're done weighing it, we're going to go back to our data analysis book and add in the weight. Now once we have that done, I'll set this down for a second. We come and we have to find the vial that matches up with ours. So here's 36. Okay. So we have our first vial for our sample. We want to carefully place our pre-weighed amount into the tube without contaminating it. Now, once we have it all in the tube, we're going to come over here and dispense 3 milliliters of methanol into the tube. Cap it and shake it to make sure all the sample is resting in the solution to extract all the fatty acids we need. And then we just put it back. And once we're done, we go ahead and we add another agent, just a very small amount in microliters, and chloroform, and let them sit overnight for 24 hours.
Hi, I am Timothy Noyes, and I'm a biology major. And what we do is on the second day, we filter it first. Then we add NaCl wash towards the tubes, which we use to clean it out. And then after we do that, we bring it over to the centrifuge, which is a big machine which basically spins at a really fast speed, so we can have separate layers. Then we bring it back here. We use a transfer pipette, which is a glass instrument, to take out some of the top layers, whatever's dirty. And then we filter it through a drying agent. Then after we filter it through a drying agent, we put it for evaporation. And then after the evaporation, we bring it over to be weighed, so we know how much of fatty acid we extract. Then after that, we add in dichloromethane. And that's the end of day 2.
On day 3, which is our longest day, we follow this protocol, and at the end of the day of transesterification, we end up with one of these small tubes filled with our solution that contains the fatty acid. And after that, we go ahead and put it in this gas chromatograph to be analyzed. And then the computer will pick up the data, and Tim will explain some of the data.
Alright, so what we're looking at here right now is the first is solvents over here. most of these are solvents. Later on, we're going to see smaller peaks, and that's going to be after half an hour or so. So this is our offline version of our data, which we've already analyzed. So what you can see here is different types of fatty acids. In particular, some of y'all might recognize these two. So we have EPA, which is commonly known as good fatty acid, and we have DHA, which, as everyone knows, is good for your brains. So this is one of the things that you can see on a gas chromatograph.
With this fatty acid research, we found that it's a very hit and miss process. When we first started, it was the first trial of any gas chromatograph technology in the forestry building. So when we were running our first processes, it was a whole episode of trial and error, and say we ran into a problem, like a lot of our vials and samples were producing salts. We wanted to minimize the error, and therefore we'd go talk to Sam Guffey, who is the lab manager here, and he'd sit down with us and help us revise our protocol in order to fix the problem that was wrong. Then, in addition to Sam Guffey, if we had any general fishery questions, we could also go to Professor Thomas Hook, and he oversees all of our interactions and decides what we do and when.